Welcome to Chapter 17, Concept 17.3. 17.3 focuses on how RNA is modified after it has been transcribed. This modification is done by various enzymes and occurs before the RNA leaves the nucleus. Keep in mind the DNA synthesizes RNA, and this is done inside the nucleus of eukaryotic organisms. RNA is then capable of leaving the nucleus, but this only happens after some modification has been done. Some of the things that are changed are the ends of the RNA strand, and some of the information is actually cut out. So it sounds like we're going to be cutting out some of the information that we transcribed. Interesting that a cell will cut out some of that information. Why would it transcribe it to begin with? So as mentioned, a few things happen to the mRNA strand. First off, I guess I should say it's pre-mRNA. To start with, we are putting a cap on both ends of the mRNA. If it's on the 5' end, it's called a 5' cap, and if it's on the 3' end, it's called a poly-A tail. Poly means many, A is referring to the nucleotide, and we can see why this would be called the poly-A tails. It's a long strand of nucleotides, nothing but A. This would be your answer to question 40, what is happening to the 5' and 3' ends. Now what's the purpose of putting on those caps and that poly-A tail? Well, let's look at number 41 here, alteration of the mRNA ends. There's a few functions that you should be aware of as to why we're adding the 5' cap and the poly-A tail. First off, they seem to facilitate the export of mRNA, meaning that it's allowing mRNA to leave the nucleus in general. No cap, no tail, stuck inside the nucleus. Also, it protects the actual genes that are contained within the RNA to be protected. There's some enzymes out inside the cytoplasm, and they could eat away at those sensitive codons, those sensitive nucleotides that create them, and the cap and the tail, they prevent that from happening. And lastly, they give the ribosomes a place to attach to, so that 5' cap, it actually allows a ribosome to quote-unquote find it and then bind to it. A second modification to the mRNA, or I should say pre-mRNA, is some splicing. So it's called RNA splicing. What happens here is, well, each cell has a different and unique function. Liver cells have liver cell functions, kidneys have kidney cell functions, lungs have lung cell functions, but they all have the same DNA. They all have the same genes. Only certain portions of genes are necessary in various cells. Hence, the selection of which sections of genes need to be expressed and which sections do not need to be expressed. We do this by cutting out only the genes of the RNA that are needed by that particular cell, and these are called introns or exons. Taking a further look at these, we can see on this image here that we have a pre-mRNA strand right here. It already has its cap and tail, but now we have sections that we are cutting out. We can see these light pink introns that we are splicing out of the pre-mRNA, forming our mRNA. The mRNA is now capable of leaving the cell, going to the ribosome to create its final product, whatever type of polypeptide that will be. Now, some cells, they'll need these introns, and they'll cut out different sections. Once again, it depends on the location of that cell, what type of things that cell does in regards to the organ that it belongs to. So to recap, exons are expressed, introns are cut out, pre-mRNA is longer than mRNA, pre-mRNA is stuck inside the nucleus of eukaryotes, mRNA will go to ribosomes in the cytoplasm for expression. Spliceosomes and SNRNPs, and the reading outline calls these SNRPs, which I guess sounds pretty good. We'll be looking at 44, 45, and 46 right here. In some cases, RNA splicing is carried out by spliceosomes. And spliceosomes are different proteins that are able to splice out introns, so to speak. SNRPs are made up of RNA, snRNA specifically, which is actually your answer to number 45, and a protein molecule, which looks like it's your answer to number 44. Spliceosomes consist of several of these SNRPs and ultimately function to determine which sections of the RNA are spliced out. In other words, which sections of the RNA are going to be introns and which are going to be left, which are going to be expressed, which are going to be those exons. They do this by recognizing specific sections of nucleotide sequences in which a SNP should be made. So here we have our pre-mRNA. Looks like this is a relatively small section. There's an exon and a second exon and an intron that we need to cut out. Going back to 44, the two parts that make up a SNRP are this protein and the snRNA. Once again, we can see that it's a single-stranded RNA strand that will be forming our SNRP. We use a SNRP and other proteins to form a spliceosome, which is specifically the item that will cut out this intron in this case. And we can see, looking at this image here, itty bitty, that the snRNA, it looks like there's some bonding going on right there and some bonding going on right there. Those will be hydrogen bonds found or formed between these mRNA or snRNA nucleotides and these mRNA nucleotides. Nucleotides form a hydrogen bond, a sequence. What that does is it allows the SNRP or spliceosome, the SNRP portion of it, to bond to it. This is actually going to be a snipping site, and we're also going to have a snipping site right here. So this is how a spliceosome can locate where specifically to cut for an intron. Once again, we don't want to screw up the reading frame of those codons. Otherwise, we're going to have some funky looking proteins produced as a result of misreading our DNA. And then after the spliceosome cuts out the intron is now a waste product. We can combine exon 1 to exon 2, which ultimately shortens the pre-mRNA to the official mRNA at this point. And the spliceosome components, they'll go away. They'll find another pre-mRNA to act upon. So looking at 46 and 47, and actually 48, number 46 is asking, how do these spliceosomes work? So think about what we just talked about and explain how these spliceosomes work. And 47 is asking you to fill in the labels for this image. And actually looking at it, number 48 is asking you to look at how these bonding sites are recognized for where a SNRP should bond. Keep in mind it's looking for complementary nucleotides that it can bond to. Same here. And that is where it will cut. So that is actually your answer number 48 as well. We also have ribozymes, which are similar in function to spliceosomes. Not exactly the exact same thing, but they have similar roles, so to speak. Ribozymes, for number 49, are catalytic RNA molecules that function as enzymes, and these two, like the spliceosomes, they can cut the RNA strands. In some organisms, the intron RNA acts as its own ribosome. So in some organisms, the intron itself is a ribozyme. It will recognize its own places where it needs to be cut, and it will cut itself out. Kind of cool. The discovery of ribozymes rendered obsolete the belief that all biological catalysts were proteins. So ribozymes, before they were actually understood, and they are enzymes, before they were understood, scientists believed that it was proteins that recognized the cutting part, not recognizing that there was actual ribozymes there that will do that. And this kind of ties into common belief back in the day when people even thought that DNA was made up of protein, when in fact it's not at all. So with ribozymes in mind, since we just looked at ribozymes, and those are enzymes of RNA, they act as an enzyme, three properties of RNA that allow it to do so. And this would be tied to number 51. It's looking for three functions. First, it can form a three-dimensional structure because it can base pair easily with itself, and we saw some examples on the splicezyme specifically where RNA can actually bond to RNA, forming those hydrogen bonds. Additionally, some bases in RNA contain functional groups. That's going way back to Chapter 2, the functional groups that have specific properties, and amino acids usually consist of those functional groups. Well, RNA sometimes will contain those as well, allowing it to have a unique function. It will do something depending on what it has or does not have. And lastly, and I actually kind of mentioned this when already, is that RNA can hydrogen bond with itself. So with other nucleic acid molecules, that would be other RNA molecules. The last section that we're going to be talking about in 17.3 covers alternative RNA splicing. Something that we should be aware of is that some genes can encode more than one kind of polypeptide, and this depends strictly on which sections of the gene is kept as an exon and which sections of a gene are trashed or which sections are introns. So because of alternative splicing, the number of different proteins an organism can produce, it's much greater than its number of genes. Once again, that's because some genes can produce multiple types of polypeptides, and these would be alternative RNA splicing.